Yep.
Good evening, one and all. I am Dr. Nilesh Dumore, FDP Co-Coordinator and Head of Chemical Department, SN Patel Institute of Technology and Research Center, Umrah, Bardoli, Surat. I would like warmly welcome to our esteemed expert speakers, Dr. Ashish Rajurkar, for today's Faculty Development Program session. So I am truly honored to have such distinguished professional with us. So it is my pleasure to introduce Dr. Ashish Rajurkar is a leading researcher in dedicated to advancing the agriculture sustainable through the innovations root system research. Currently leading the field research component of the harvesting plant initiative HI, HPI at Salk Institute as a senior research scientist. Dr. Ashish spearheaded here the large-scale field-based phenotyping efforts across the major crops, including rice, maize, soybeans, canolas, vital crops for the agriculture. With extensive experience in international and multidisciplinary collaborations, and Dr. Ashish has worked alongside leading the institutes such as the International Rice Research Institute, IRRI, Philippines, Duke University, University of Illinois, Urban Champions, and University of Missouri's in USA, and International Means Weeds Improvement Centers in the Mexico. Their research integrate the cutting-edge techniques like salvinomics, root corings, advanced imaging to explore how the root trend influence crop productivities and crop resilience. And dedicated the scientists and mentors, Dr. Ashish is the committed to bridges the research with real-world agriculture solutions. Their expertise in phenotyping, field-based experiment, and global collaboration place them at forefront of developing climate smart crop that contribute to food security and environmental sustainability. This is all about our the today's expert, Dr. Ashish Rajurkar. Now I will enjoy the healthy session with Dr. Ashish sir. So I request to Dr. Ashish to continue his sessions. So please, Ashish sir, you can continue.
Yeah, please. Yeah, thank you, Nilesh, for kind introduction. I think let's try first. I think who is admitting means I am getting all admit notification. So
Yeah,
can we
right.
cover that one? Okay. And let's try. I think sharing first if it is worse or okay. Can you see my screen or how it is?
No, it not.
Okay, hold on. Able to see my screen now? Hold on. Share.
No, no.
Okay. Yeah, I can now. And then allow. Okay, you able to see my screen now?
Mm,
Yeah, it looks like you are sharing my means it is only my this one, but I can share. I think.
no, no, it not.
How about
It's blank.
now? Okay.
It's blank white
Not the
screen.
PPT.
No, no,
Okay.
it's not showing PPT. It's only white page.
share screen. How about now?
Uh, Dr. Ashish, please enter the screen.
Yeah, it's full screen already.
Mm, then it's still not showing.
Okay. How about hold on? I think I can join on my this because this app I don't know, but I can join with the website one. Okay. Yeah.
Okay. Otherwise, you can send PPT to us.
Yeah.
Uh, we can
It's
directly
big
present it.
PPT. Hold on. I think I will join from their website, so it will come.
Okay.
Hello. Can you hear me now?
Yes.
Yeah. Okay. Let me try from here now if I can share. Okay. Now you can see my screen probably.
Yes, it's visible now.
Okay. But I can't see the people now. How to go? Okay. View. Speaker. Okay. So can you see my screen?
Yes.
Yeah. I don't know because I can't see people. Anything I can see my slides only.
Okay, no issue.
Okay. But if it is not visible at or it is interrupted at some point, please let me know and
Yeah, sure, sure. Don't
we
worry.
can hear. Yeah. Yeah. And then I think yeah. And is it moving? Slides are moving.
Mm, it's
Yeah.
showing first slide. Yeah,
Yeah.
it's
Now
moving.
next.
Outline.
Okay. Yeah. Okay. Good. Mm, okay. Now is it still screen is visible?
Yeah, it's visible. First slide.
Okay.
Reading
Okay.
Resilience Foodcraft.
Okay. Okay. Good. Okay. Hello everyone. So shall we start?
Yeah, sure.
Yeah. Yeah. Okay. So hello everyone. Sorry I can't be there in the India, but currently I'm in USA and it's morning time here. But for you all it is evening time. But hope you are enjoying the sessions with Dr. Nilesh and Dr. Chandra. Um, so let's get started. I think so. Already Dr. Nilesh has introduced me, but to brief, I'm Ashish. I work as a senior research scientist at the Salk Institute. You may be knowing Salk Institute. Many people knows, but Salk Institute the polio vaccine. But all of us know polio vaccine. So the person who developed the polio vaccine it is Jonas Salk, and he has institute in San Diego. They work on all human diseases, including cancer, HIV, and all other things. But we have some plant division. We have small plant division. There we have one initiative called as Harnessing Plant Initiative, where we leverage the capacity of plants to sequester more carbon. So I work there, and you can reach out to any of the social media platform with me. You can email me, you can WhatsApp me. This is Indian number only, so for WhatsApp, and you could reach out on the Twitter or LinkedIn. So let's get started. Basically, I will be talking about only few things today, not much. But I divided that talk into three major outlines. One, first of all, I will take about the food security challenges, what challenges we have that I will be describing. Then, with that one case study with the root traits for the drought resilience, specifically in rice, and then followed by the root traits for soil improvement or healthier soils, with the example of the soybeans. And mostly, I will cover in food security. I will cover about global climate change. What are the issues? How we can mitigate it? Some of the solutions, and then I will be coming with the more detail with the case study in the rice with the drought in the rain-fed condition, and then soybeans with the diverse population, how we can leverage the diverse population for understanding roots, identifying candidate genes, and how we can improve the soil health. So let's get started. I think, as of all of you know, this Oxfam, it is one collaborative organization of the 17 institutes or more than, and then we know we celebrate World Food Day on 16th October 2020. But their mission statement of this Oxfam, who works on the livelihood or poverty and food security concerns, or equity concerns, and they says every person or should have enough food. So their statement is, we believe in a fairer world with enough food for everyone. And today we are more determined than ever to make it happen. So with that one, I would like to tribute Dr. M.S. Swaminathan, who was father of Green Revolution in India. Um, mm, Unfortunately, he is no more with us. He just passed last year. But I met him during World Food Prize ceremony in 2014, and 2016 also. And this was one of the picture from him. So before going more into detail, I think I just want to start with this video from the NASA, uh, and learn about the what is climate change. So this was the video from the NASA, which was predicting how our climate will be in the future. But if we see in the backward, like what was there in the previous one. So this is another short video I put here from 1880, how is the temperature rising, and you could see that one with this graphical illustrations in the right hand side. So all this is the mean temperature change within means each year, and you can see the how the temperature is changing or how temperature is rising over the years. You can still hear me, right? And these are the recent years. We could see how the temperature is changing and it totally turned into the red. And this is from 1880 to the 2022. And to summarize this one, this is the graph I put on the left side, and you can see almost from the 1980s we got like temperature increase, but this last recent 9 years these were the warmest in the earth. So how this temperature again it affecting the our crops and other things that I will be telling later on. But there are really climate change issues which are much more concerning, and we need to mitigate those one. Apart from that one, we have population is also growing so fast, like exponentially. In 2012 we have 7 plus billion population. In 2050 we uh, predict it will be 9 plus billion, and in 2100 it will be more than 10 plus billion population, which actually needs more food to feed them. And their food production actually should be increased by 60% to feed this 9%, 9 plus billion people in 2050. But as you look into our land area or earth surface area, we all know there is a 71% water and only 29% of the land. And in that 29% of the land we have to make this means all land it don't go for the only for the food production or agriculture. So only 38% of that land, 29% is comes for the agriculture, and then 62% land we use for the other, like we have homes, we have other offices, those covers in that land. In that 38% of agriculture land, only 33% is the arable land, means we can produce food in those lands, while 67% are the marginal lands. So it is really less land, marginal land, which is like unproductive land. But we have only 33% arable land, and with that 33% arable land we have to feed these 9 plus billion people in 2050. So there will be really less land per capita um, to produce the food. So there will be, to summarize this, there will be more mouths to feed in a insufficient arable and marginal lands. To summarize all this global climate, to summarize that video also, and then temperature increase. If you see in 1950s there was a really less temperature. In the future there is a temperature increase. We are almost in 2024, we can see the temperature increase already, where there is more carbon dioxide level also. So those are all got increased. There is a more temperature, more carbon dioxide, there will be less water, and we have more populations in the coming years. So for to sustain this one we need sustainable solutions, which can you can find on the right side. So we need to decrease this temperature, we need to reduce this carbon dioxide by some means, and we need sustainable solutions to cope up with this climate change. So With that one, I told you now what are the food security challenges, what are the what how the climate looks, how these are predictions will be there, how population is increasing, and how much area in agriculture we have, and what are the solutions for it, to cope up with the climate change. So to start with this one, I have the first case study during my PhD studies in the rice and drought, where we targeted root traits for the getting resilient cultivars that I will be going through. So as you know, so temperature is one of the stress. Similar drought, there will be less water in the future, and there will be more abiotic stresses in the coming year. For example, this from the plot on the figure on the left side, you could see all these red area mark in a mark on the map. Those will have severe drought in the coming decades, in the 2014, and this is actually a major constraint for crop production in rainfed ecosystems, specifically in India or all over the world. And if this drought occurs at a reproductive stage, like when the grain gets peeled, it reduces really yield by almost 50%. um, uh, it reduces. And so to cope up with that one, we need to develop a drought tolerance cultivars. And now if we want to develop a drought tolerant cultivar, we need to understand first mechanism of the drought tolerance. So how actually we can, if we know mechanism of doing something, then we can play with that mechanism. But if we don't know how is the mechanism, then we can't play. So if we look into the mechanism of drought tolerance in the plant, basically it has two mechanisms, maybe escape or avoidance or tolerance. In the escape mechanism, what plants do, plants basically they want to complete their growth cycles, and then it means early, and then they want to escape the drought stress. Or if we have another mechanism called as avoidance or tolerance, they can develop some secondary traits. For example, they can have high stomatal conduction, high osmotic adjustment, or high transpiration efficiency. They can delay their leaf senescence, or they might have deep root systems, which I will be talking more in the latter point. And they can extract more water, or they have some adaptive traits which are response which are triggered by the drought stress. But most of the I will be talking about root system today. And all of you know, roots are important for maintaining crop yields, especially when plants are grown in a soils with insufficient water and nutrients, because roots are main source which can capture the nutrients and water from the soil and make plant uh, to survive. So basically, in India or all over the world, mostly we have some elite or cultivars which we use for the food production. Like if you are eating some food or eating some rice, this is from the elite cultivar like IR20. But there are some locally adapted land races, or which are, we can say, stress tolerant genotypes, which are there in the world. But in Tamil Nadu, where I did my PhD in Tamil Nadu Agriculture University, we have these locally adapted land races called as the Nutripata, Nurungan, or Kalandukar, which are actually having deeper root systems. These land races usually have really low yield, but they have some resistance mechanism in that one. And you can see these land races usually have are the reservoirs of useful genes, and they endow tremendous genetic variability, contributing to major QTLs or genes in aquatic stress tolerance. So we utilize these land races to make further studies, and we have some elite cultivars also, like they which are yielding high. This like IR62266, it is from ERI maybe, and then CT999 3. This is high yielding varieties, but if you see IR62266 and CT999 3 in the picture, this IR62266, it is green still, means we gave imposed the stress to the plant, then little we gave really less water, and then after 21 days we could see what are the differences in these plants. And you can see there is this IR62266 is still green, meaning it has more osmotic adjustment, while CT993, which we already known for the it is upland adapted japonica, it is stress tolerance also. These are all both are stress tolerance, but they have different mechanisms. So CT993 it is deeper and thicker root systems, while IR62266 it has a higher osmotic adjustment. So with these populations, actually, these Norunga, Nutripattu, these are the land races, and these are the light cultivars. We develop some mapping populations, which we call recombinant inbred line, and we have some near isogenic lines also um, developed within IR20 and CT999 to the non-agriculture background people. These are just we are if we want to study the genes or genetics, we need to have some mapping populations like offsprings of those one, and then we can study how these traits are translating in the future generations. Nils are basically towards developing variety, but these are the mostly like backcross lines. So now if I have those lines, I have some crossings made and I have those lines, but I need to evaluate them in the target area where actually drought occur. So if you look into the map of the India, we have Tamil Nadu here on the just the southernmost state with the white color, and then followed by the Kerala in the down one. Okay, so we all know we get rain. For example, in Gujarat, we get monsoon rain. So monsoons come from this Kerala, and then it goes towards Gujarat sides, and then after in the Gujarat and then Rajasthan desert, it again this monsoon gets returned towards again back in the towards Tamil Nadu side. So that will be so we called it the southwest, and then or south and southwest, and then west to then again um, down west south. So the return monsoon basically they get in this part of the Tamil Nadu. So this is more focus in the Tamil Nadu now. So we know all Rameshwaram and all, but near to Rameshwaram we have these districts, Rameshwaram districts, we call as the Paramkudi, Ramnad. This district basically they get at the return monsoon, which coming from the um, back from the deserts. And usually we know return monsoons are really weaker; they don't give that much rainfall. And in that little rainfall, farmers in this area, specifically in the Paramkudi, Rameshwaram, Ramnad, they want to grow the rice. But it is obvious there will be less rain, and in that small rain, they want to get some rice yield. And basically, you could see they do some direct seeded rice sowing, where this farmer in the picture they just they sow the seeds, rice seeds in the barren means like open field, and they just wait for the rain to come. So when rain comes, this seedling get emerge, and then you have some crops. But I evaluated those mapping population in that target environment for 3 years almost. So in 2012 and 13, you could see I means we planted around 20 September, 25th September. There was a rain, and then plants were emerged out, and then was little rain. But after that, there was no rain throughout the cropping season, which resulted in the severe drought in that year. Similarly, in 2013-14, what happened? First, initially only one rain came, and then seeds got emerged out. But later on, there was a really less rain. Only 217 mm rainfall was there during that year, and you could see from the picture, this all soil has cracks and all. Mostly you have seen soil this rice in a paddy fields where water is stagnating and farmers are growing. But this is just a dry seeded and direct seeded rice, and you could see in such conditions it has to yield. So it needs really potential. While 2015 was normal year, it has much more rainfall. So in this within this population, actually in our lab earlier, we mapped some QTL with using the IR20 and Nutripath background for the grain yield under drought, or several physiological traits, including root volume, root numbers at one of the location in chromosome number one. So this location then during my PhD studies, I find map into the smaller region, and I find out it is related with the grain yield in the under drought. And this is the well-known region actually with the QDTY1. So yield QTL for the under drought, and it is linkage with the SD1. This we published in the 2019. You could read that paper. Similarly, using another population, I have mapped consistent yield QTLs or yield under drought QTLs with the another population, which was IR62266 into Norungan. And I have found out consistent yield QTLs, which are earlier reported also by different users from the International Rice Research Institute, and we published this one in 2022 21. So now coming to the next uh, mm, population, we have these near isogenic lines. As I showed CT999 and IR62266 in earlier slide, where IR62266 was green in color, having high osmotic adjustment. But CT999 it has, I told you, it is deeper root system or thicker root systems, and they have high penetration ability also. Like this is in the wax layer. If you put and then they can go high penetration, they can penetrate. So this is the one thing. It happens when stress occurs, the soil gets really hard, and if you have that ability of the plant, so it can grow more, it can penetrate those hard soils, and it can extract the water from the deeper layers. So we have population near isogenic lines developed from these two parents. We use basically um, some QTLs which are related to thickness, root thickness, or root pulling force, or penetrated root thickness on chromosome number 4. And we have developed near isogenic lines from that by the backcross method. So this is just a standardized protocol of the development of the nails. You have IR20. IR20, as of you all know, this IR20 is the higher yielding uh, rice line, where which came in during the green revolution time. This is basically short, and you can see the root system of that one. It is a really shallow root system, and while CT999 it is really thick root system and deeper root system, and we develop these near isogenic lines, nils, which are really thicker and deeper root systems. These nils then I have evaluated in our target environment in the Paramkudi, and you could see in 2014 I told you it is really stress year, and these nils have all the thicker root system and deeper root system, while 2015 it was normal year mostly, and they have performance is normal performance. So mostly when stress comes, they will increase their rooting ability and they can grow the deeper. Okay, and these lines also we evaluated for many years in that area, for example 2010 to 2015 16, and we found those lines have consistent higher yield. Each year is different actually. There may be already less rain, high rain. So for example 2013-14 I told you, but these some nils they showed higher yield compared to the either of the parent. Similar, they have another mechanism also. They can flower earlier, so they can escape the stress, and then That mechanism also we seen in those near isogenic lines. And obvious these nails they were developed for the root system, so they have deeper roots, or they have more number of they have thicker root system, um, compared to the either of the parent. So to then we thought, oh, this is really good genetic region, which we can interrogate further into some different elite cultivars, because farmer now don't grow IR20, they grow new higher yielding lines. So we thought, okay, we could interrogate that region into some of the elite cultivars in like ADT 37, ADT 43, ADT 45 from the Tamil Nadu, and that program also I did earlier during my PhD studies. But now we can see from these markers, you can there is some background differences in all near isogenic lines. For example, if you see blue color, they have more of the city background, either of the parent, and then if you see red color, they have the IR20 background. And according to that only, again, that also affects their uh, performance. For example, I told you city 9993, it is a stress tolerant, and has a deeper and thicker root system. So with that one, those near isogenic lines, we were knowing, oh, they are performing good, they are growing good in the drought stress in the target environment. So to study what is actually the mechanism of those drought, those lines, I took those lines in the International Rice Research Institute in the Philippines, where we studied them into the well water and stress condition. You could see this picture from the well water, and it's on the left side you have well water, on the right side you have the drought stress one, and in the little zoom version you can see it has um, stress given. But this is not the natural stress, this we manage, this is the managed stress during reproductive growth stages. So we just gave less water, or we shut down the water irrigation during reproductive stage, which resulted into reproductive stage drought. And I could see the differences in the root systems in those near isogenic lines against at the International Rice Research Institute. Mostly all the lines which we have has a higher steel diameter, which is resulted into the higher thicker roots compared to the IR20 or CT 9993 in reproductive stage drought. Yeah, again, those lines in the International Rice Research Institute also, some of lines they perform better than IR20 or IR20 or CT 9993. IR20 was really performed better there because IR20 is adapted to Erie environment already. It is from the Erie itself and it perform in its native environment. Now, as you all know, these roots are really hidden into the ground. If you want to study them, it is really laborious, time consuming. You can't dig all the roots. So I was looking like, oh, what is the again their architecture? How they um, grow into the soil, or what makes them stress tolerant? To understand that one, I have collaborated with the Duke University, and then there they have this system called as a gel-based system, where we could evaluate all our near isogenic lines with the parent in this gel-based system. And you could see, and I was able to impose the stress with the PEG mediated stress under these gel systems. And you can see these are the non stress, this is non-stress condition. These plants looks really healthy. There are more leaves and really vigorous roots, while in the stress there are few roots, and uh, there is less leaf area and all. And interestingly, uh, in the non-stress versus stress condition, I found these near isogenic lines has a thicker root system compared to the non-stress condition. You could see that one from the graph also. So all the near isogenic lines, they have more thicker root system compared to the IR20 in the orange color in the down in the stress condition. And you can see that one also from the picture here. Now, that was first we studied physiological mechanism, we studied root system architecture. Now we need to go towards genes more. Which genes are the actually responsible in those near isogenic lines? And nils are the best material to study those one. So what we did again in the greenhouse condition, we grow them in the PVC pipes which are really like 1 meter long, and we impose stress and the non-stress condition. You could see this is the IR20, it is already drying in the stress, but CT triple nine still it is green, and all our nils they also are green. And you could see those near isogenic lines have in this one in right side picture. You could see this is the one is the non-stress and one is the stress, from the color itself, and these near isogenic lines, for example 212, 172, 29, they have deeper roots in the drought stress, similar to CT triple nine three, while IR20 is basically shallower root systems. And we found most of the genes which were responsible or which were responsible for the stress tolerance, and those were the upregulated genes in those near isogenic lines. And it is clear from this one, for example this CT triple nine three, which is a drought tolerant. This is the control versus stress. Yes is the stress condition. In the stress they have very less differentially expressed genes. Similar in this 297 also, you can see the both are the blue color, means there are very less differentially expressed genes. While this another nil, they have some differentially expressed genes. Um, but this 297 showed really less differentially expressed, which may be a promising, and it has much more city background, and could be a promising cultivar in the future years. So I told you now up to about root traits from for the drought tolerance in rice. We utilize land recesses, and for the deeper we identified granule cuteals under drought, and we have some developed some near isogenic lines which were performing better in a drought stress, and we find out some differentially expressed genes which are responsible for drought stress tolerance. So those differentially expressed genes we are validating in the future, but these lines are already in the advanced stage of their performance, and they will be, I think, released in a few years in Tamil Nadu. Coming with that one, I would like to go to our next topic, where what I am doing currently. So we are targeting root traits for sequestering more carbon into the soil. Basically, I will be telling story from the soybean today, not other crops. We work on many major crops. But to start for that one, I would like to go again to the another video. Um, this is from the NASA Earth Observatory, and it can you can see CO2 emissions from the Earth, how much CO2 we are emitting and how it changes across the season. So this is again from the 2020, or and then you could see rising carbon dioxide in the season. This is world map, and you could see all these oranges. These are the CO2 emissions from those lands. So basically, when crops are there, there is a less emission of CO2. While when the crops are during October, now this is the harvesting time basically. So when crops are not there, there will be more CO2 emission in the environment. While when the crops are there in the field, there will be less emission of carbon dioxide. But it is obvious it is changing too much, and we are almost now 450, around 500 ppm CO2 level is there, which will again increase in the future years. Again, this northern hemisphere and southern hemisphere, they will have changes because cropping pattern is different, because one get winter, one get then um, a normal years. So it will be alternate, so that you could see clearly from this video how the changes in the northern hemisphere and southern hemisphere occurs. So to take away, it's like carbon dioxide level is increasing, and it will changes with the cropping season also. So, and we could see when the crops are there, it was carbon dioxide level was really down in that area. So with that objective, actually, currently at the Salk Institute, we have initiative called as Harnessing Plant Initiative, where we want to leverage the plant's natural ability to capture the carbon dioxide. But if we are capturing that carbon dioxide, where we should be putting into the ground, into the soil, by making some bigger roots or deeper roots. So just we can put some polymers like some of the lignin or suberin into the roots, which are already naturally there, but we can make their abundance more, and then they can be little bit stays there for a longer duration. So basically, in the Salk Institute, as a harnessing plant initiative, we are doing we are making plants, Salk ideal plants, so called, with the deeper and bigger roots, to sequester more carbon from the environment to the soil. And then soil, if we put more carbon into the soil, soil gets healthier, and it can be used for another, again, as a crop, for the crop growth, as a fertilizer. So I told you, means plants already captures carbon dioxide, and if you see the statistics now, so human also, the activities like we drive car and we do many things, so that also results into around 37 gigatons of carbon dioxide released by humans, and around 727 gigatons it is released naturally. So out of this one, only 746 gigatons can be captured naturally. So we have some excess now, 727 plus 37 if you calculate, and then if you minus from 740, you have around 18 more gigatons, which is excess into the atmosphere, and which can be a problem in the future. So if we target to capture these 18 more gigatons by developing some plants with the bigger roots, or suberin rich roots, it can sequester almost 20 to 46% of the this one. It is not the full solution, but it can be a part of solution, and to the capture the carbon. With that one, basically, we want to solve climate crisis by deeper and larger roots, and we target major all the major crops. Now in the agricultural land, if you see which are the crops they are grown more, for example, cereals always rice, maize, wheat, these are the cereals, you can see they are major crops growing all over the world. Similar, then coarse grain, maybe barley or sorghum, or some oil crops like soybean, canola, cotton. These are the crops, they are the major occupies the major area all over the world. So we are targeting all these crops, for example, wheat, maize, rice, soybeans, sorghum, then canola, but specifically I work on the soybeans and canola at the Salk Institute. So I will be talking about soybean today then. So if we now we need to have some, you can say, how you can say this is like, if you have some, you need to find some genes then, right? So those are already present in the environment with the like genetic diversity. So we are basically leveraging that genetic diversity to identify candidate genes in the soybean. So we have one population, diverse population of the soybean, which came from all the different parts of the world, and this is highly sequenced. Almost 5 million SNPs are there, and we leverage that one and for the to study the root system architecture. And you could say this is the diversity in the soybean population that we have. This is from our collaborator, where they studied the roots on the paper based system and then they captured the root system architecture. But in our lab, what we did, we have similar this jail based system what I told you during my Duke study. So this is just a cylinder, these are the smaller systems where we can grow the plants, and then we can study the root system architecture of those plants. And with the cylinder based system, this is gel based system again, and we can capture their architecture, and then we can study many different traits. So with this population and this system, we did some genome wide association studies and identified some candidate loci, which I can show in the future. But this paper we have currently on the bioarchive. If anyone is interested, they can find it and they can read it out. I can share also in the future. So with this system, we found one gene in the soybean where if that gene is there, it can have shallow rooted system or low tip depth, how much, and or the high tip depth. If it's high tip depth, then it is going the steeper. Basically, we want to grow the roots bigger and then deeper. So why deeper, I can tell you in the latter slides. But for the deeper, if we want to grow in the deeper, the basic idea is if the roots architecture, these roots are growing steeper, then they can go in the deeper. So steeper angle roots, they go deeper. That's the takeaway. And we found this one gene basically in the soybean which has a steeper root angle. And this again, this gene already it was published in the from our lab, same lab itself, in the Arabidopsis, where if you have one allele of that gene, it can have shallow rooted system, or if you have another allele, then you have a deeper root system. And then currently I'm leveraging that one and then working on this gene to uh, develop a deeper root system. So with that one, I was also interested in studying like natural accessions, or the diverse population, at least subset of the population, how they actually relates to the carbon sequestration. And then we have this is the map of USA, and I have four different sites across USA. Basically, now if you see, San Diego is on this side towards Pacific Ocean, and much more desert area is there, but many productive lands are in the east side, where this is the we call as a Midwest. This is the productive lands of USA, and I have four different sites there where I have taken field trials. I will tell about one field trial at the University of Illinois, I guess. So now I told you how to study the roots in the different system, from gel-based system or digging roots. But in the soybean, we could leverage this taproot system by doing a shovel mix. So shovel mix is just basically a shovel where that shovel we use to dig the roots, and then you can get these clods of the roots totally. You could wash those roots, and these wash roots we have developed some system to image them, and you could get this image of the roots. And then after getting that image with the computational pipeline, you could analyze those images to extract the bunch of traits, like how much root length is there, or how big these roots are, how these thicker roots are. Those you can get. These are the different traits you can extract from those images. And this is one genotype. Consider it is in the all four different location, because there will be always environmental interaction to grow in the for the root systems. So that's why was four different um, sites, and you could see performance of one genotype across these four different sites. And interestingly, from that one, we were able to see if we capture those images, and then we can extract volume as the one trait, which was actually giving us strong correlation with the physical mass of those roots. So we don't need to get then physical weight of that one. We can calculate the volume from the image, and we can say, oh, this has this much mass. And it is already well known from earlier studies in a maze. Volume is the best predictor for cloud root mass. So, as I told you, volume is one of the predictor, but we want to grow them into the steeper root, like we want to grow them into the deeper layers. So, what is the matter? It is just the angle, how the angle of the roots is there, and if you have steep angle, they will grow more deeper, while if you have shallow angle, they will grow more in the shallow layers. And we could see the steeper genotypes have more roots in the deeper layers. From this is another modality we use. We took root cores from the soil, and then we wash roots, and then we could see how many roots are there in different depths. So this is, if you see, deep means roots below 30 centimeter, below 60 centimeter, or below 90 centimeter. There is a strong positive correlation from this average orientation. So if you have more orientation, means it is the steeper, and it has a more roots in the deeper depth. So we could confidently say, like, oh, steeper root growth, they have more deeper roots. And this is clear from that, from the this plot itself also. For example, this genotype, this is yellow in color or green in color, for example E300. This is also a landrace, which has a steeper root, and has a deeper root growth. Now, how this relates with the your carbon change? If you have deeper, you are putting roots into the soil. Can we see some changes into the soil carbon? And this is just a preliminary data, but you can see some changes, a little carbon change from time point before planting versus after planting. Some carbon change is there in the deeper layers. Mostly deeper layers is the we could see changes more because we are putting roots there. Now, this is, I told you, I showed you how we are making the deeper roots or bigger roots, and then how we are putting them into the soil to make it the carbon, and I can see the little carbon changes there. But if you see now, why we want to put them into the deeper layers rather than the top layer? So we could call that as the topsoil versus subsoil. So if you have deeper roots, they will obviously grow in the subsoils, while in the topsoil, the shallow roots will grow. Most of the all the cultivars in the recent years, they have shallower root systems. Now, if you look into this left side panel, in the topsoil, mostly all the processes occurs. Okay. For example, if you have plant growing shallower, or you do tillage, there will be all the processes occurs only in the topsoil layer. So there will be more microorganism, there will be more oxygen availability, so activity will be more. And then carbon, though carbon will get here more carbon with the more roots at the topsoil, it will not stay there for the longer duration. while exactly opposite in the deeper in the subsoil layers. If you put roots in the deeper layers, there will be less oxygen, there will be less microbes, there will be all the processes are slow in the slow or less in the subsoils, and which will help in putting that carbon for longer duration. And it is clear from this one of the study where they studied these different different plants, for example, grasslands, croplands, forest, and they could see in the top centimeter you have much more, almost 80% of the roots are there, 70 to 80%, and only 20% of the roots are there in the deeper soils. But still, if you see the residence time from this middle plot, the deeper layers one, they have more years of the residence times. So deeper from this one, takeaway is just if you put roots grow deeper and then put carbon in the deeper layers, it can stay for the longer duration. But obvious, this soil organic carbon is a complex trait, and it is affected by uh, many things. For example, soil chemistry, like how soil pH is there, how is the soil quality is there, which quality of the soil is there, which enzymes are there, and recently which are the microorganisms are there, what they do. So again, microorganisms also use the carbon in the soil, how much they use, all these factors, it is they affect the soil organic carbon, and it is complex, but we are studying those all the parameters, and we'll have um, results in the coming years. So from this one, from the whole presentation, just takeaway messages are roots are often underestimated but hold immense potential for crop resilience. There should be enormous diversity in crop root system, and which we should utilize those one. Then breeding for resilient root traits will help in a drought resistance and soil health improvement. I could say from the carbon studies, subsoils have large potential to sequester more carbon, and improvement of a deep rooting traits will be beneficial. Um, Also, but to understand all those one, we need better understand those systems, and relationship between soil carbon and their other related traits, how it affects the soil organic carbon. And breeding resilient food crops with optimal root traits will ensure sustainable agriculture. That is, I am sure about that one. So with that one, I would like to thanks all my mentors, all my collaborators from different parts of the world. And basically, this is the picture from the Tamil Nadu. These are the helpers who helped me during my PhD studies. Uh, though I was not knowing their language, but I was learning more from them. And with that one, thank you all, thank you for listening. Yeah, I'm happy to have some few questions.
Yeah, thank you, Dr. Ashish.
Yeah,
Today, there are two questions on chat box. So I have
mhm.
just read it out.
Yes, please.
What are the thoughts on the water cost of the cultivating paddies compared to
Mhm.
other crops, and any suggestion for the varieties that required minimal water?
Yeah, so that's good question. So most of all of us know rice is grown in a paddy fields. Paddy is the, like a stagnating water is there, and it has much more. So water is the high, or rice is the high water requirement crop, you can say. And if you have paddy rice, that will obviously it is increasing in your cost for the water with the irrigation and all. But alternative technologies like recently they are coming, for example, we alternate wet and dry. So something like you give some water and then release that water, when again plant need, again you can irrigate that one. So that will be our one sustainable solution. You can't directly go for the mm, direct one, but their alternate wetting and drying, those are the sustainable practices they are doing. And there are now universities, they have cultivar, especially International Rice Research Institute also has some varieties recently released. Those are specifically for those technologies. You could look specifically in that their website to get the specific genotypes.
Okay, one more questions. How can one improve
Mhm.
germination rate of soybean seeds?
Oh wow! I think yeah. Um, so basically, what we do, that is same problem we are facing in US also. If we have recently harvested soybean seeds, we definitely can get good germination. But what we found, I think if you give just hot treatment, means if it is really like which say you are doing, if it is in the field, um, I means use the fresh seeds, basically fresh means from the last year harvest or something. But we found like if we give some hot treatment before germination in the field or in the greenhouse condition also, like put in the oven or sun dry it for 1 day, and then it helps in a germination.
Okay. So any more questions from participant? So please drop on to chat box. Otherwise we go for further process. Any questions? I think no more questions from participant. So we will go
Yeah,
further.
please reach out to any of the social media platform. I'm happy to answer anytime.
Okay, thank you, sir. So now we'll go further. Thank you very much, Dr. Ashish, for giving very informative session with us and for providing very important informations about the food security challenge, root threats for the broad resilience in the rice, and root threats for the healthiest soil in the soybean. So all this information is very important to our participant. So I hope information is provided by you. It will be useful and applicable to our participants. And thank you very much for accepting our invitation with very short period and providing us with a very your time.